No, you'll be fine. He's
Somebody else on the
Yeah.
curve is a little.
I brought a translator. I'm okay.
That's right.
So we looked at the DNA repair genes on cytotoxicity of gemcitabine. So gemcitabine is a pancreatic cancer drug, the most common one that we use, and so it causes DNA damage. So we want to look at what genes are responsible for fixing that damage that it causes. And so a little background information. That's gemcitabine. So it's a nucleoside analog, which means it basically looks like one of the 8 T's and G's that make up our DNA. More specifically, it looks like cytosine, and so the only difference with these F's instead of H's. And so what it does is it can trick your DNA polymerase into incorporating that instead of the normal bases. And so once it incorporates that, then the DNA polymerase is kind of like, uh-oh, I just did something wrong. But it's actually really unique in the fact that like there's other nucleoside analogs that kind of act in the same way, but as soon as those are incorporated, they just kind of like halt transcription, and like the DNA polymerase can remove it with its proofreading activity and everything's fine. But gemcitabine is actually unique in that once it gets incorporated, it can incorporate one more normal nucleotide like right afterwards. And so since there's that normal nucleotide like right afterwards, the DNA polymerase's proofreading activity can't remove the gemcitabine, and so that makes it resistant to the proofreading activity. So that's kind of how it causes the DNA damage. It'll stop chain elongation, and so yeah, that's kind of the background. Again, pancreatic cancer is like one of the most deadly cancers. Usually by the time you start showing signs, it's already too late. So that's kind of a hot topic in cancer research. And so there's actually, patients will become like more resistant to the drug as like they're getting it, or they might just be like born resistant to it. So that's why we're also trying to look at these what genes play a part in fixing this damage. And so hopefully we can make that drug more efficient, lead to longer prognosis and stuff. And so we did like 3 parts in our research. And so part 1, there's some previous research done that says, so XPF is a protein that can come in and cut the DNA. And so we think that that's responsible, like in one of these pathways, repairing this gemcitabine-induced damage. And so, like our previous research showed that when XPF is inhibited, all of a sudden these cells are way more sensitive to gemcitabine. So XPF is obviously doing something in this repair of gemcitabine-induced damage. So there's another group that also looked at another endonuclease called APE, and it kind of showed the same traits. So we wanted to see if these 2 endonucleases act in the same pathway or different pathways. And so that's what this is showing right here. So we suppressed both of these proteins with siRNAs, which is kind of cool. I can tell you about that later if you want. But so if they're in the same pathway, I'll tell you if they're in different pathways. So we have XPF pathway and APE pathway of like fixing this gemcitabine-induced damage. So if we knock out XPF, we still have this APE pathway. So like our cell survival should still be fairly okay. But if they're in the same pathway, like if you knock out both, well then, or just one, like, then, you know, it's kind of not great. And so we suppress one or the other, and then both in these 2 different cell lines. We have our HeLa cells, and then the pancreatic cancer cell line. And so we found that like suppressing both endonucleases actually has like pretty similar effects of just suppressing one or the other. So that makes us think that they're in the same pathway, which is kind of exciting in the fact of trying to target these proteins eventually someday. And so, I mean, you can kind of see, yeah. But anyways, that was kind of part 1. So part 2, a little more background explaining. So this gemcitabine will cause a single-strand break in the DNA, and that, after the next round of replication, will be a double-strand break. And so there's 2 ways of fixing double-strand DNA breaks. There's the emergency way called non-homologous end joining, where it's kind of just like, and just smashes them back together. And then there's like the more perfect way, where like it uses the other chromosome as the template, and then it leads to like, you don't have any mutations or anything. And that's called homologous recombination. And so you would think if you had had a defect in homologous recombination, that you would be more sensitive to gemcitabine, but because you don't have that ability to fix double-stranded breaks perfectly. But it's actually the opposite in pancreatic cancer cells, so that's kind of weird. So what we did is we proposed this hypothesis of actually how gemcitabine induces these, like how it induces cell death in homologous recombination. And so we think that the DNA polymerases that are involved in homologous recombination, they'll be going along making their new nucleotides, and then they come across this gemcitabine and they're like, oh no, what is this? And so they'll try to like use their proofreading activity to get rid of it, but it can't because of this masked chain termination is what we call it, the extra nucleotide. And so it'll spend extra time on the DNA trying to like combat this little gemcitabine, and that in itself will block the other repair proteins from coming in to actually fix it. And so that's kind of our hypothesis of how we think homologous recombination like causes cell death. And so what we did is we have two cell lines, one that's BRCA2 proficient and one that's BRCA2 deficient. And BRCA2 plays a big role in homologous recombination, so if you don't have BRCA2, then you're not going to be able to do homologous recombination very well. So with the previous studies, like the ones without BRCA2 can't do homologous recombination, so they actually will be more resistant to gemcitabine because you need homologous recombination to be like killed by gemcitabine. So these are our two cell lines. CAPN1 is the BRCA2 deficient, doesn't have BRCA2, so they should be resistant to gemcitabine. And then the CAPN1-C2-1, those are BRCA2 proficient, so those actually are more sensitive to gemcitabine. And so what we did is we exposed these two cell lines for different amounts of time to gemcitabine, and then looked at the presence of DNA polymerase epsilon, DNA polymerase delta, and then a couple other proteins that are involved. And so what we found in the BRCA2 proficient cells that can do homologous recombination, the DNA polymerases were expressed more. So that's kind of good news. Like they might be binding on the DNA longer, trying to combat this gemcitabine, which is what our hypothesis is trying to say. And so, I mean, that's kind of exciting as well. And another little thing over here, we also exposed them to the same two cell lines to another DNA damaging agent called mitomycin C. And so mitomycin C also causes double-strand breaks, but they use, it's kind of the flipped from gemcitabine. Like the ones that can't do homologous recombination are more sensitive, and then the ones that can like are resistant. So it's really weird. It serves as a good control because they have opposite effects on like the cytotoxicity. And so like we didn't really see really a difference in the polymerases in either of the cell lines, which is also kind of good news because that means something funky is going on for gemcitabine. So it's something special, some pathway, something different from mitomycin C that's going on. And so that's kind of that. And last but not least, we are trying to create, so there's kind of multiple ways to suppress proteins in cells. Like we use the siRNA, but sometimes the siRNA doesn't always work, like because you have to get that siRNA all the way into the cell, and then it has to associate with one protein and then another protein, and then it could do its job. But sometimes it doesn't always work with different cell lines. So what we're trying to do is use this CRISPR-Cas9 technology. So what CRISPR stands for is clustered, regularly interspaced palindromic repeats. So basically in the DNA you have these little sections that are palindromes that are repeated, and they're interspaced and clustered. And so this is kind of discovered in bacteria first. This is how bacteria get their immunity to foreign pathogens like viruses and stuff. So say a virus injects its DNA into the bacteria. Well, the bacteria will cut up that viral DNA and take a little chunk of it and like insert it in between these clustered chunks of DNA, and so then the next time around, when that virus comes in, it can it has that little chunk in its DNA. It'll go and transcribe that to a little RNA, and then that'll go associate with this Cas9 protein, and that'll become like this entity, and then so that can go find the viral DNA and then chop it up, and then everything's good, like the cell's alive. So that's kind of what we're doing in these cell lines. We have these little RNAs that are complementary to either BRCA2 or the XPF gene, and so we can incorporate that into a cell like with the Cas9 protein. So hopefully that'll go find that spot on the gene that's homologous and then cut the gene in half, hopefully making the gene like not work. And so if we can eventually like create an entire cell line where these cells don't have BRCA2 or don't have XPF, that would make things so much easier in the world of like doing experiments, because like you wouldn't have to deal with the transfection efficiency is what we call it, of trying to get the siRNA into the cell. It might work, it might not, which totally throws off everything else. But and so that's kind of like part 3 is we're still in the process of that, but we do show some signs of optimism where each one of these are different little gRNAs, and you can obviously see some suppression of XPF in these 3, because their bands really aren't even there. So that's definitely like optimistic. And so, and then this one's not really as obvious, little subtle suppression of BRCA2 right here, but again, hopefully those will be better with further incubation with the CRISPR-Cas9. And so yeah, future, I guess basically to sum up, if we can figure out like what fixes the damage caused by gemcitabine, then we can ultimately start to make more personalized medicine. And because we find that like if we know what fixes the damage, if we stop those from fixing the damage, then those cells die more, which is good. I mean, that's what we want. And then yeah, that's basically what we found in a nutshell. So, it's a lot of background knowledge, but try to do my best.
A lot of teachers.
We know that gemcitabine is not used only in pancreatic cancer.
Right,
it's breast cancer, ovarian cancer, a variety of other things. So when you start talking about that creation of cell lines, you cross into the Human Genome Project. So we know that cell tracing and we know that cell manipulation is acceptable in breast cancer because people have wrapped themselves around that. It hasn't even been talked about in pancreatic cancer. How do you cross the ethical barrier?
That's a good question. You might have to repeat that for me. So.
Okay, so we know that in women's studies, women's health, that gets a lot of attention. We know that women are doing elective mastectomies. We know that they're doing...
It's based on their genome.
Right, we're doing family history studies to say, do I have the genetic predisposition to get breast cancer? Right. So I want to immediately start early treatment of that. That's acceptable in society because it's women's health.
Right.
And as part of the human genome study, we haven't crossed over that into pancreatic cancer because we view pancreatic cancer as a male disease, not so much as a female disease. How do you get people to give it as much attention as we are in women's studies?
And so I would say to start off with the fact that pancreatic cancer just has such a poor prognosis. Like by the time you find out, it's usually already too late. And like literally like the 5-month survival rate is like less than 5%. It's ridiculous. And so what this part 2 found, the fact that we need homologous recombination for gemcitabine to even work, that in of itself is important, because if you have a defect in a homologous recombination gene somewhere in your genome that you can pick up, you're not going to benefit from gemcitabine at all. So you're only going to be harmed by its side effects. So that's important to know.
So you can give them something else.
Right, exactly.
Wasting that 5 months on a drug that we know won't work.
Right, we don't have 5 months to, and so, and then also, I guess, obviously this is still very early in the process of, but once we do figure out I don't know what genes are responsible. That'll lead us to be able to screen more, and I don't know. But as far as
early
it's
predictor,
as big as breast cancer, this is going to take some time, I guess. Where once we find out more about it, get more evidence-based studies showing that hey, like such and such actually works, then we'll probably be able to get it on the radar. Because while it's not a super like popular cancer, it is deadly. So.
Do you think that part of the lack of detection is just like it used to be with stomach cancer? It was, oh, you have ulcers. So they treated you for ulcers forever, only to realize you really had stomach cancer.
Exactly.
Pancreatic cancer, they treat as pancreatitis, a stitch in your side, all those different things are really a precursor to lupus. And by the time they actually get to figuring out it's not that, it's cancer, that 5 months is gone.
Exactly, yeah.
So do you think it's because they're not thinking of that first, they're thinking of it last?
It's tough to say. I mean, just the fact that the signs don't really show themselves. So you could be walking around with pancreatic cancer for a few months and not even know it. And then by the time you start feeling like, something's wrong, like, it's, physiologically too late for us to really do anything. It's already metastasized. It's already, and so that's
There's not the staging like there
exactly.
is.
Yeah, it's tough. It's a lot of gray area, but hopefully this will help. And there's a lot of research being done. Even here, like a lot of kids are doing pancreatic cancer stuff. So definitely a widely researched industry as well.
Would you do it again?
Do this again? Yeah, of course. They're even saying since I'll be a junior next year, I could come back next summer and do it again. One of the other kids in our cancer, there's 5 of us in the cancer department. This is his second year, and so definitely something to consider. It's opened my eyes where I don't know if I really want to go like MD PhD, but I definitely want to be involved in research, like eventually somewhere down the line. And so, I wouldn't. I thought before this program, I was like, no, research, that's not for me. Like, you don't need it to get to med school. Like, okay, I probably won't do it. But after doing this, it's pretty, especially cancer research. That's why I want to focus on this.
Good.
Awesome. Good job.
Awesome.